Hello, welcome everyone, and thank you very much for attending the first Human Cell Atlas Middle East Symposium. We will start shortly. All right, welcome everyone again. So my name is Youssef Idaghdour. I'm a faculty in the biology program at New York University, Abu Dhabi in the United Arab Emirates. So I would like to start by thanking the Human Cell Atlas team, and in particular, Alex Shalik and his team. The team engaged with us and with colleagues in the Middle East, as part of the effort of the HCA to drive inclusive representation and participation in the HCA initiative, basically globally. And I would like to also thank all members of the advisory committee who helped us in the last several months in preparation of this event, as well as future events that we are planning to organize. So we have an exciting set of talks today and tomorrow by a group of leaders in the field of single-cell genomics, as well as young scientists and students from the Middle East. So just a quick thought about the Middle East and genomic initiatives. So countries in the Middle East have been traditionally underrepresented in genomic projects, including genome-wide association studies, and single-cell genomics is not an exception. So the field itself is an emerging field in the region, and the Human Cell Atlas, as a highly collaborative project driven by the community, is really an opportunity for us scientists in the region, not just in the Middle East, but in the MENA region, in the extended region, to really start actively engaging in such collaborative initiatives. So again, today and tomorrow, we have an exciting program, including keynotes, and we also have 6 sessions in the program covering topics from computational data science to experimental and technological advances in the field. And importantly, many talks highlighting single-cell research in the Middle East, as well as panel discussions about engagements and empowering this type of science in the Middle East. I just would like to mention that this is an HCA meeting, and we have a code of conduct that all participants should adhere to. So the link to the code is shared in the chat. So without further ado, we are really lucky to have Sarah Teichmann as our first speaker. So Sarah is the head of cellular genetics and senior group leader at the Wellcome Sanger Institute. She's a co-founder of the Human Cell Atlas with Aviv Regev in 2016. And so Sarah continues as a principal leader of these initiatives, which really keeps expanding, really as a driver of collaboration, training, and sharing of data sets and resources in the field of single-cell genomics. Sarah is an MBO member, and she is also a fellow of the Academy of Medical Sciences, the Royal Society, and as well as a fellow of the International Society of Computational Biology. So her work has really been driving the field in so many ways, and this work has been recognized by a number of prizes. So Sarah, thank you so much for presenting the Human Cell Atlas, and please proceed with your talk.
Thank you so much, Youssef, for that kind introduction, and also for spearheading this first Human Cell Atlas meeting, Middle East, which is just fantastic, and I'm so pleased that that we have this meeting going on, and grateful to everybody for joining today. Can you see my slides properly?
Yes.
Yeah. Yes. Fantastic. So welcome everybody. And thanks again, everybody, for joining. So the Human Cell Atlas is, of course, as Youssef said, it's a grassroots community effort to create a comprehensive reference map of the types and properties of all human cells, the fundamental units of life. And we're doing this project as a basis for understanding our own biology, and then also for translating it for the benefit of diagnosis, monitoring, treating disease. What unites us in addition to that mission of mapping the cells in our body is are our principles and values. Oops, sorry, something's happened with my camera, sorry. I'm just going to stop sharing for 1 second and restart the video. Apologies there. Is it working now? Sorry about the technical glitch. Screen share. Microsoft PowerPoint. Can you see this now?
Yes.
Is this okay? Yeah. So anyway, so to say that really what we are committed to and emphasizing is both the quality of the project, the data, the results, flexibility, community, equity, diversity, and inclusion, both with respect to the community, but also the samples, transparency and open data sharing, privacy and ethics, and then technological innovation and excellence, both in terms of the experimental techniques and computational methods. And of course, the goal is essentially to make a periodic table of cells in terms of the constituents of our tissues and how they work together to make the tissues and ultimately the organs and our physiology. And the way we drive that project is through the methods, the techniques in the bottom right, and the way we organize ourselves is also in terms of bio-networks that I'll come back to later, which are shown on the bottom left. So in terms of the sort of history of the project, Youssef already mentioned that we had a kickoff meeting in 2016 with about 100 participants, kind of on the left of the timeline. And, you know, now, of course, we've built up the community through working groups, bio-networks, meetings. You know, we've had milestones in terms of cells analyzed, and we've now grown to many members across the world. And we're really at a juncture in the project where we're actually integrating data to make gold standard data objects for individual organs and tissues, like, for instance, the lung. And we're at the point where we're generating multi-tissue and also multimodal atlases with both suspension and spatial components. In terms of meetings, you know, we've always had general meetings on an annual basis, and then we've expanded to also regional meetings, meetings with a focus on equity, joint meetings with other partners, like, for instance, the NIH, COVID symposia. We've had some nice meetings with focuses on particular biological topics like development and pediatric. And this year you can see we've had also regional meetings and workshops. The HCA Asia was the first regional meeting right from the beginning. And of course now, you know, we're delighted that we've got this HCA Middle East Symposium, which is mostly virtual, of course, but I guess there are people who are having sort of regional hubs with meeting rooms and group showings, which I think is an awesome idea. And, you know, a lot of fun and a great way to do things. So the way, because, you know, we wanted to have a really international grassroots community from the beginning, you know, it's very gratifying that we're now, our membership includes people from, you can see, many countries and all inhabited continents on the world. And we stand at over 3,000 members, which is really amazing. We've had also, of course, with so many members, a rapid growth in the output of the project, sort of covering the cells in our body systematically over the years. And you can kind of see an exponential growth, both in terms of the number of individual donors and patients who've given samples, the number of samples, and also the number of cells, which is getting close to 100 million in terms of suspension cell data points, which is really awesome. As I mentioned, kind of the COVID pandemic really spurred on the formation of biological networks, because we realized that we have to have experts working together for all the different tissues and organs, starting with the lung, which was, because COVID was viewed as primarily a respiratory disease. And now we've got 18 different biological networks, including ones like organoid and genetic diversity, but also the different systems and organs. And these communities are really focused around people who are interested in those specific systems. And then, because we're kind of coming together in terms of phase 1, like a draft human cell atlas, and we're at the beginning of entering into phase 2, and I'll talk more about that. What we've also encouraged is basically for each of the biological networks to develop a roadmap of where we're going forwards to comprehensively map each of those tissues and systems. And you can see here 5 of already published roadmaps, which is fantastic, because it then helps the community to kind of focus on the path forwards in terms of what are the different tissues that need to be covered, how do we want to generate data and analyze them, and so on. It's also worth mentioning that we have, in addition to the biological networks, from the beginning, we had working groups on analysis and equity and ethics and standards and technology. They were formed really early on, and they provide communities for addressing each of these areas. And in terms of governance, it's also worth mentioning that Aviv and I co-chair the organizing committee, which is kind of the deciding kind of hub, basically, for coordinating the whole project. And we also have a data ecosystem oversight group that provides kind of steering and coordination and some governance around the data storage, and the data portal and analysis and so on. So in terms of the analysis working group, this is really focused on computational methods. And the group has also organized meetings, jamborees, and so on over the years. It's focused on querying the atlas, how the atlas is actually assembled, how it's used, and also includes community tools like the cell annotation platform. The standards and technology working group has the remit of comparing, benchmarking, disseminating methods, providing guidelines, best practices, discussing, the new technologies within the group, and identifying areas for improvement. The ethics working group, so Youssef has already mentioned Alex Shalek. So Alex Shalek, Musam Lang, and Partha Majumdar coordinate this working group, which was formed right early on in the project, because we really wanted to have this principle of equity kind of as a foundation of our community. And it's been amazing in terms of promoting, supporting progress, engaging across biological sexes, ages, ethnicities, geographies, socioeconomic status, empowering participation, outreach, education, and training. And they've also written several pieces, as you can see there. We also have an ethics working group, and this is really for participant information sheets, consents, ethics for donor and patient samples, and there's a toolkit online, basically, for helping guidance, kind of in terms of general principles internationally for these legal ethics governance, kind of around patient samples, which is, of course, really important. I want to highlight that we have a publications website where you can submit your manuscript as a human cell atlas publication. There's a website and an email. It's a very simple process to just submit a Google form. And what's key is in the acknowledgement section to add this publication as part of the human cell atlas, and to post the publication on bioRxiv so that it's linked. And it will then, the benefit will be that you'll have exposure. The data will enter the human cell atlas, the paper will gain visibility, highlights, the contribution to HCA will be on our landing page, and can also have social media kind of exposure. Great. So I've given basically an overview of the project. I want to speak a little bit now about basically, how do we benefit from having a map of the cells of our body in terms of the complete molecular fingerprint at single cell resolution, and just give a few vignettes of the science of the Human Cell Atlas over the years. And I'm going to start with an exemplar. We're having even just a draft of major tissues and preliminary cells, and it gives us a molecular guidebook to our own bodies, and that can be incredibly powerful, for instance, in terms of mapping viral entry factors to understand infection routes. And so as I mentioned, the pandemic really spurred on the formation of the lung biological network, and because we were able to assemble data from across our tissues, we were the community, it was amazing basically how quickly we all came together to share data, not only published, but also pre-publication, and then map where the viral entry factors ACE2, TMPRSS2, and so on were expressed in the cells in our body. And what that pointed to was conjunctival epithelial cells, in the eye, kind of nasal epithelial cells in the nose, and later also ductal cell epithelial cells in salivary glands in the oral cavity, that all gave us clues as to the sites of transmission, human to human transmission at this interface. And in fact, we published this in April 2020 in Nature Medicine, and that work then also guided public health policies around mask wearing and routes of transmission. And of course, then there are also information about internal, deeper down in the lung, and internal cell types that have the viral entry factors and transmission inside the body and so on. That guidebook concept, I just want to say, that guidebook concept in terms of the molecule of high precision, sort of Google Street Map precision of the molecules and in terms of cell location in the body, is obviously also useful for understanding genetics and drugs, and I'll come on to that later. And so in terms of the Human Cell Atlas as a molecular guidebook, we can also use it to understand where drugs are acting. And so the cardiac conduction system that we published earlier in the year is an example of that. So this conduction system consists of pacemaker cells, sorry, I'm flipping back and forth, that you can see highlighted in the sinoatrial node, which is one of the tissues of the cardiac conduction system. The pacemaker cells are amongst the only spontaneously firing cells in our body, and they determine the rate at which our heart beats, kind of from basically the early development of the heart to sort of when we take our last breath and it ends, kind of those cells are spontaneously firing, many dozens of times a minute, and are kind of get also information from the sympathetic and parasympathetic innervation of the sinoatrial node. So these cells are kind of really crucial to our body, and of course, knowing the molecular fingerprint of the human pacemaker cell is then useful for understanding how drugs are affecting either the speeding up or the slowing down of our heart rate by acting on those cells in a direct manner, or potentially indirectly. But the point here is that having the molecular fingerprint of those cells told us, for instance, that they have the GLP-1 receptor expressed, potentially on their cell surface. And of course, that's the target of liraglutide, semaglutide, which are marketed as Wegovy and Ozempic, which are these diabetes and obesity drugs. And indeed, those drugs have a 6 beats per minute reported side effect on increase in heart rate. And so what this Human Cell Atlas data is telling us is that effect could be potentially direct through this expression of this receptor, rather than some sort of indirect effect. So it sort of guides us in terms of understanding how drugs are acting on target in terms of intended and unintended effects. It also basically helps us to provide potential mechanisms at the cell and molecular level for genetic variants. And so one example are genetic variants that have been reported as leading to variations in digit number or length. So brachydactylies, syndactylies, and so on. And, mapping the cell atlas of limb development and hand and foot plates and digit development basically informs us about precisely when and where those genetic variants could be acting. So in summary, basically this human cell atlas has a utility in terms of being a guidebook, both for pathogen entry factors, for drugs, where drugs could be acting even in the healthy reference atlas, and where genetic variants could be acting. So the other little utility basically that I want to highlight is that the human cell atlas sort of gives us the blueprint or the recipe also for engineering cells in the dish based on developmental pathways and tissue microenvironments that we see in the in vivo human cell atlas data. So for instance, one example of this is T cell development in the thymus. The thymus is the little immunological organ next to the heart where T cells develop, and knowing the molecular features of that developmental pathway and the tissue niches where this takes place, from the cortex to the medulla of the thymus, can inform us on how to engineer T cells from iPS cells in artificial thymic organoids, because we then know which transcription factors, which signals, and so on are needed in vivo, and can leverage that for engineering in vitro. So the human cell atlas essentially also gives us that blueprint for cell engineering. Okay, so in just in the last few minutes, I'm going to talk about key challenges that we face at the moment in the community. One is atlas integration. I'm not going to talk about that for a long time because John and Lucia will talk about the data integration efforts that are going on in the community at the moment. And I've already said that the cell annotation platform is in development now and led by Chloe Villani and her and collaborators and team, which is a centralized platform that can be used for harmonizing and seeing different annotations of cells from different groups. So ahead, in terms of also those roadmaps that I mentioned, we want to expand the atlas in terms of tissues covered and in terms of modalities, as well as human diversity. And of course, what we need then, once we have all that data, is to be able to query the atlas and have computational and data science approaches and AI/ML methods that allow us to learn and query, foundational cross-body models. And some of them out in the literature are shown here: SCGPT, GeneFormer, Similarity. So moving from annotation to function is kind of the final challenge for the community ahead. So for instance, we can describe and map cells, but it doesn't necessarily give us all the facets of function. That's, in a sense, understanding the function of every single cell and characterizing is beyond the remit of the human cell itself, but we'll partner basically, the human cell sort of provides the foundation, and the map is the framework for partnering with other projects where we are looking at perturbations of that healthy reference data of the cells. And the disease is the most obvious one, genetics is another one, and then obviously in vitro perturbations is another one. And with that, I would like to end and just say a huge thank you to the entire human cells community, and of course also to Youssef and the committee and the team that's made this possible, which is really exciting and fantastic, and happy to take questions.
Thank you very much, Sarah. So for our audience, if you have any questions, just post them on the Q&A. And probably could start with a question. So whenever we think about the value of diversity, it's very clear when we think about genetics. But when it comes to actually functional data, has there been ideas about how to add value through inclusion, for example, of patients or individuals from like diverse genetic backgrounds?
Yeah, absolutely. Absolutely. So I think that the human cell atlas community isn't like the remit for mapping, all of human genetics is not kind of part of the main aim initially. So you can sort of think of the human cell as like the Human Genome Project. So we need a reference to start with that we can then map genetic diversity onto, like GWAS would then be the next stage, for instance. And so that, but that's sort of ultimately, the roadmap. And for instance, International Common Disease Alliance and so on are using cell atlas technologies for then, expanding to lots of diseases at the genetic level. But the human cell is really the starting point for that, I would say. And of course, we want to have a reference that is useful in terms of covering, the some global diversity component in order to provide the reference for those bigger efforts. But it's the whole of like cell atlases of the whole global genetic diversity in the whole world is a really big project, and that I would say would be kind of the next stage. But of course, it's already starting in parallel. Your point about function is key, and I completely agree that having the genetic perturbations and seeing how cells and tissues change with genetic variants, that is a key way to unravel function. It's one way. It's one kind of entry point. And it's really powerful, of course, because we can there's only so many ways that you can artificially perturb human body. So you rely, genetics is brilliant, and really important.
There is a question on the QIA related to diversity as well, I think.
Oh, yeah, I see it. A question regarding diversity. Are we expecting to have unique type in different healthy populations? Does that mean unique cell types, maybe? Are we expecting to have unique cell types in different healthy populations? That's a good question. I mean, my hunch is that there wouldn't necessarily be, or there could be, it depends on how you define cell type, but certainly, in different healthy populations, there'll be different variants of cells for sure. And that's partly driven genetically, but it's partly also driven by the environment, right? So for instance, the immune system is very plastic with respect to exposure to different challenges. And so yes, we would expect some unique features or aspects of cells, let's say, in different healthy populations. Maybe not entirely unique cell types, but different variants and states. So there are more. So how do you want me to do this, Youssef?
Yeah, maybe in order. So
just in order. Okay, so Kate Spacey is asking how many cells are required to see HCA 1.0. I guess, in the, I didn't emphasize it a lot, but in the white paper that we wrote right at the beginning of the project, we said that a draft atlas would include, around 100 million cells from all the major tissues and organs of the body. And then we had a priority list of about a dozen major internal tissues. So that's where we're headed. There's a lot of detail in that's in the white paper. And of course, you'll hear more about our integration efforts and where the community is working towards from John and Lucia. So this next question from Ragad is, how do you account for or integrate the heterogeneity between donors, especially when increasing the diversity of HCA donors? Okay, so that's also a great question. And I think, again, Lucia and John will talk more about this in their talk on integration. There's a component there that's the challenge is basically how to integrate different techniques and regress out technical and technical differences, but maintain the heterogeneity that's biological and inherent. And I won't go into that in detail, basically, out of interest in time, but you'll be hearing talks from the computational community here, Molad Falahi, Lala Hadverdi, and other people in the program who will talk about the computational integration techniques, which are obviously key to the whole project. Then the next one, Shaksada is saying, thank you for your talk. How will the single cell data be integrated with arising epigenetic single cell data? Okay, that's another integration question, which I'm sure we'll hear more about in the talks. And essentially, it depends, the computational techniques depend on whether it's joint data or separate data. And there's a whole class of linkage and integration techniques that are based on different principles of statistical approaches. But in essence, the epigenetic data just gives more features per cell. So with the expression data, you've got about 25,000 features per cell. With the epigenetic, you may have 100,000 or however many peaks there are in the open chromatin. And so from a statistical point of view, it's not that different. It's simply more features per cell. That's a whole area of research that's very exciting and ongoing now. So yeah, thank you everybody for your wonderful questions. Thank you, Youssef, for introducing and sharing. And
thank you so much, Sarah.
No,
I appreciate it. So next we're gonna move to the next talk, so by John Randell and Lucia Robson. So John will introduce Lucia next.
Thank you very much, Youssef. I'm John Randell from the HCA Executive Office. I'm here with my colleague, Lucia Robson. We're very glad to be here, very glad to have an opportunity to expand on what Sarah has already introduced about how atlases are actually going to be assembled and are being assembled in the context of the Human Cell Atlas Consortium. I should say at the beginning, the HCA Executive Office consists of about 13 professional staff who are based in 4 countries around the world. That includes Singapore, Japan, the United States, and the United Kingdom. And collectively, we're responsible for a wide variety of tasks in support of the global consortium. That includes the organization of meetings such as this one, the coordination of the biological networks, project management for various components of Atlas assembly, and so forth. And so it's really, our goal is to really provide a very strong support that enables the science to be done, and that enables particularly the collaborative science that HCA is based on to really run smoothly. So I'll lead off by expanding a little bit on some of what Sarah has already introduced about HCA and the timeline and the process of Atlas assembly, and then I'll turn over to Lucia to go into more detail about the actual process of Atlas assembly. As you've already heard, we are really sort of in a new phase of HCA currently. The first phase, which really began with our founding in 2016, was centered around data generation, community establishment, protocol development, pipeline development, and so forth. And you've heard about some of these timelines, the establishment of our working groups in 2018, the very intense work on COVID that began and really was published at the very beginning of the pandemic in 2020. And then beginning last year is really when we entered into this concerted effort to assemble atlases that then collectively will become part of the ultimate human cell atlas. The first of those, as you've already heard, was published in June of this year. That was the lung atlas. And then recently, we've also published actually 2 nervous system atlases last month in Science Magazine. And as I'll describe in a bit, the strategy that we're taking is to take a tissue-by-tissue approach to generate a large number of atlases that then can be combined to form the first draft of HCA, which will be available by 2025. That is only the first draft, however, and so following HCA version 1, there will be many iterations going into the future where we expand the data available, the data types that are available, and really work towards getting to a comprehensive atlas. So I want to stress that even this intense phase that we're in right now of assembling atlases is really just the first atlas assembly step, and the product is just the first draft of the Human Cell Atlas. So what does that mean? Well, as you've already heard, this work is really based around the HCA biological networks. There are 18 of these biological networks. They're represented here on this slide. Each one of these networks in this current phase of HCA is creating one or more atlases that will then be combined again to form the full atlas. In some cases, these will be whole tissue atlases, for example, the lung. In other cases, there are actually sub-atlases. So for example, the first atlas from the eye network will actually focus solely on the retina. In the case of the immune system, we're focusing initially on the circulating immune system, and we'll go on to tissue-resident immune cells at a later date. And so, so again, the product of this first phase is the first draft. And so if you can go to the next slide, I want to talk about what we really mean by the first draft. And we already had a really excellent question in Sarah's portion of the program on how many cells will make up the first draft. And so to review, the white paper imagined that there would be about 50 to 100 million cells making up the first draft. But because it is a draft, this is not going to be a comprehensive atlas. As is represented by this middle panel, there will be gaps in, there'll be anatomical gaps, there will be gaps in particular tissues where we have not covered all cell types in that tissue in this draft. And as you've heard both Youssef and Sarah talk about, there will be gaps in genetic diversity, in ancestral diversity. And so those gaps and more really will need to be filled as we move from the first draft of the atlas to a more comprehensive human cell atlas. I do want to say a little bit more about diversity. This has really been the goal since day one of HCA, is to really be a globally representative atlas. And so meetings such as this one and the HCA regional networks are a fundamental part of our strategy to diversifying the atlas and going beyond this sort of historical focus on particular populations that Youssef mentioned at the very beginning of the meeting. There are many aspects of human diversity. Obviously, ancestral diversity is a major aspect, but you have gender, you have experience, geography can affect transcriptional profiles, and so many different axes of human diversity that we need to take into account in order to build a diverse atlas. Now, why do we need a diverse atlas? Well, that's a really critical attribute of any atlas that is going to equitably benefit humanity. And so the ability to understand disease, and particularly sort of regional variations in disease, it really rests on having an atlas that is representative of all of humanity. And that's why HCA is putting so much emphasis and so much effort into diversifying our atlas. So I hope that we can have more discussion about this aspect of HCA throughout the meeting. But right now, I'd like to turn the program over to Lucia, who will go into more detail about the actual process of combining data sets to create the atlas. And I just want to say at the beginning, in order to appreciate the scale of this, I would just point out that even the very first draft of HCA requires the combination of anywhere from 300 to 400 individual data sets from all around the world, covering all the tissues that you've heard about already. So this is really quite a large scale project, and that is really the challenge that we're facing. So Lucia, please proceed.
Thanks, John. It's so nice to be here this morning. Thank you for all joining. I fully echo John's sentiment about kind of the complexity and the scale. It's really hard to get your head around, but looking forward to kind of walking through the practicalities of atlas assembly in the next sort of 10-15 minutes. So you've already heard so far that we're having versions of the human cell atlas, and version 1 will be a really great start, but it's certainly not the end point. Some of the features of version 1 that I wanted to highlight today is that it's really, first and foremost, a healthy reference. The priority is to create a healthy benchmark from which disease and sort of other elements can be mapped on top. Another feature is the leadership of the biological networks. So we'll keep coming back to this group. They're a really important group. So there's 18 biological networks, each representing kind of organs, tissues, and systems. And in order to develop atlases that are meaningful and biologically relevant for each community, the biological networks and their coordinators are really kind of the liaison point to create atlases that are meaningful for these groups. We've already spoken a little bit about diversity thus far, and John just reiterated the goal of creating a representative atlas is a really ambitious one, but we have to start somewhere, and we're certainly really committed to this. So There are teams, and hopefully we'll hear about them throughout the rest of the meeting, which are really focused on how do we measure diversity. So even if we can't yet achieve kind of a full representative atlas, which is a huge ambitious goal, we really need to be able to set ourselves up to kind of measure incremental change towards a more diverse and more representative human cell atlas over time. So the sort of infrastructure around how we measure diversity is being set up at the moment. A fourth feature of version 1 is a commitment to refining the methods that we use to assemble atlases. Obviously, atlas assembly is something that's only kind of come up in the last 5-10 years, and more recently, I guess the rate of change and the improvements in the methodologies used computationally is really blowing everyone's socks off. So the team is quite determined to take an iterative approach and test and tailor different methodologies over time. So it's certainly not a one-size-fits-all, and especially at this stage where atlas technology is really improving all the time, the commitment is to kind of continue to refine methods. So that's a little bit about the features of version 1 rather, but we can't forget versions 2, 3, and 4. And some of the features of those include a greater commitment to capturing diversity. Obviously, we've just spoken about. Spatial data is just on the horizon. Everyone's really excited about that. We're already thinking about how we can include that into our atlases going forward. Other multi-omic data, also another avenue we're really excited about. And just as you can imagine, the scale will increase exponentially. So you have the volume of cells, but also the different cell types, and also anatomical coverage will increase as we have latter versions of the atlas. So I'm going to try and briefly explain the process of assembling atlases. As you can imagine, this is quite an iterative process. Essentially, I'm trying to condense this into a single slide, but essentially there's 3 phases. Up front, there's around data collection, defining the scope of atlases, getting the team together. Then you move on to a technical phase where you test a number of integration benchmarks or different methodologies to find the one that doesn't over or undercorrect. Then you create your integrated object, and in phase 3 is kind of about drawing out the biological insights. So holding community cell annotations, and then obviously the downstream analysis. And the important thing here is to say that this isn't a one-size-fits-all approach. It has to be tailored, it has to be nuanced, and it has to be iterative with the help of the biological networks. So who is actually constructing atlases? We have a global team. Obviously, the organizing committee, which is the governance group that Sarah first mentioned, helps set the strategic direction. The biological coordinators and the champions are really the link between the atlas builders and the communities that the atlases will be useful for. There's a project integration team, which are the actual postdocs in labs around the world who are doing the nuts and bolts integration, and the integration team is generously supported by the Chan-Zuckerberg Initiative. And finally, and kind of fundamentally, it's the HCA community who are providing data sets and providing input and the kind of computational as well as biological direction to build atlases that are meaningful and that are useful for the community, and grateful for them and their contribution. So we're really pleased. I'm glad that this meeting has fallen when it has, because we can now say that we have 2 atlases that have been published. The lung biological network was in June in Nature Medicine, and more recently, the nervous system biological network, partnering with BICAN, have brain atlases that were also in Nature. So highly recommend everyone have a look at them. And as you can imagine, the methodology will evolve over time, but having 2 atlases already published is a huge achievement, and we're looking forward to the subsequent ones. Okay, so once we have an atlas, where on earth can people find it? We have a centralized HCA data portal, which has recently been redesigned. It's quite intuitive to use. If, for example, you're interested in looking at the lung atlas, you click on the little lung icon here, and and you're taken to a page whereby you can see the atlases, you can explore them, you can download the data, you have all the contact information, you have the code, you have the publication, and then a second tab, you have all the constituent data sets that kind of feed into these atlases. So it's really transparent, it's free and available for everyone to use, and I highly encourage everyone to have a look at the new data portal, which is just being launched. Another piece of infrastructure that I wanted to highlight is the cell annotation platform, and this platform could have an hour's talk just on the features of the platform. So I'm not going to do it justice, but essentially it's a really nice platform whereby people can upload their data sets, and in public or in private settings, you can annotate. And why this is so important for Atlas assembly methods, or Atlas assembly efforts rather, is that once we have the atlases, we want to kind of come together as a global community and build consensus cell annotations, and that all can happen in CAP. So in 2024, we very much welcome everyone on the call to participate in global jamborees and other cell annotation efforts, which will all be run and managed through CAP. We're flying through, but I guess the last message I really wanted to share with the community today is we would really, really value your involvement. The quickest and easiest way that you can get involved is to join a biological network. Please use the QR code, otherwise you can join on our website. And essentially that's your way into HCA. You can contribute data, which we are endlessly grateful for. You can support Atlas assembly. You can bring your computational or biological or even kind of coordination expertise to help these atlases be built. We really encourage people to participate in the cell annotation jamborees, which will be happening in 2024. As being part of the biological network, you're able to join meetings and webinars and really collaborate and kind of expand your networks, which will hopefully kind of result in more opportunities for collaboration and fundraising and brilliant science. So the 18 biological networks are listed on the bottom of the screen. And with that, I'm going to stop, and I really welcome any questions. I should have given everyone a heads up before to put your questions in the Q&A. We can wait a minute. Please add your questions, but if not, John and I are certainly reachable through the HCA website or through the team that's organised this event today. We'll be happy to take any questions after the call. Okay. John, do you want to final remarks?
Sure, I can wrap up. I was going to suggest maybe, do you want to talk about, we've talked about the 18 biological networks. Do you maybe want to mention which are sort of up next in terms of what are we currently working on?
Sure thing. You're going to test my knowledge because I don't have a list of networks up in front of me. But essentially, as you can imagine, tackling all 18 Atlas assemblies would be logistically, more than anything, a real challenge. So what we decided is to divide each of the 18 networks into 3 groups. They're affectionately called waves. So there's biological networks that are in wave 1, wave 2, and wave 3. Wave 1 started last year, and those efforts have been going for about a year. And I'm going to miss one, but you can imagine that there is the retina, immune, kidney, brain, gut, and You can imagine that there's a second 6 which have just started, and they're about a year behind the first wave. And so at the moment, those 6 biological networks are reaching out to their communities, collecting data sets, getting ready for ingestion, defining the scope of their atlases, defining kind of the biological questions that they'd like to answer, and kind of really nuancing the methodology that I showed before, so it makes sense for their community. And then we can, in 2024, we'll start the final 6, which will benefit from all the learnings from the first 12. And those communities, I shouldn't say that they're not working. They're definitely busy working away now, collecting data sets, engaging with their communities, kind of preparing for the integration effort, which will commence next year. Okay. Thank you, everyone. Please don't hesitate to reach out if you have any first questions. Otherwise, I'd really like to hand over to Ido Amit, who will be giving the next talk. So thank you so much, and let me hand it over to Ido.
Thank you so much, John and Lucia. So our next speaker is Ido Amit from the Weizmann Institute of Science. So just a brief introduction. So Ido is a professor of immunology and one of the leading experts in single-cell genomics, working on a wide range of applications from like immunology to cancer immunotherapies and development, brain, and probably most importantly, the work he started was really kind of driving the technological advances in the field. I would like to actually mention that the first single-cell transcriptomics talk that I attended myself was actually delivered by Ido in 2014. And if I do remember, it was like single-cell data from few thousand cells using fax. So I think the field came a long way in the last 10 years. So we're really fortunate to have Ido because, again, he has this very interesting overview, not just of the most recent advances, but also of how the field evolved, because again, he was one of the early adopters of single-cell genomics. So Ido.
Thank you, Youssef, for the introduction, for the beautiful introduction. And I'm really happy that influenced you on with our data back in the days. And it's very tragic how this event coincides with such a horrifying events happening in the Middle East. And I think this, we need more of the connection, more of the good things that science can do and people can do together. And I hope our next Middle East event will be something that we share together, some great connection and good things that will happen across this beautiful region that can have so much and do so much good. And I hope it will come from this devastating event to grow something beautiful in the future. So I want to kind of share today, in the time I have, 3 different bits of information, which may be, I think, or may be helpful to think about some aspects of the Human Cell Atlas. So obviously, I think we're growing in the capacity to generate very large atlases across many different modalities, and together with the powerful new analytical approaches, we're discovering a lot more than we had known in the past, and this really stimulates a lot of new both research as well as development in the biotech arena. One area that we are quite excited about, and I think is really important, is the many different autoimmune diseases, especially the rare one. And there's people assume more than 100 different autoimmune diseases, and for many of them, we have very little information. And I'm going to just showcase one, which is called the scleroderma, which is just a disastrous fibrosis, autoimmune disease causing a really devastating effect to women, typically ages of 30 to 50. And the more we work on this arena, the more we are passionate about understanding the disease and around finding some type of medication that would actually have an effect. And the whole research was a brainchild of Professor Hamutal Gur from Hadassah Medical Center, and actually working with a lot of Palestinian doctors and patients from different places. I'll show them at the end. So anyway, there are a lot of clinical challenges associated to the disease. We really don't understand what are the drivers. We don't have good biomarkers, and obviously therapies are not really working at all, especially all the ones that working quite effectively for other autoimmune diseases. And there are many research challenges that I don't have time to go into right now. And then what we started doing is working together with all the physicians here in Israel, as well as the one that work with the Palestinian authorities, and then sampling both the skin biopsies as well as the blood of both healthy donors as well as patients in different stages and different disease stage, early disease stages as well as late disease stages and different manifestation of the disease. Collecting as well all the relevant metadata, and then going through sequencing of the cells, as well as the RNA and chromatin and so on and so forth. So this is constantly growing. Now we have more than 150 patients and so on. And what we've discovered, to our surprise, is the very dramatic changes that are occurring both in the various fibroblast population, parasite population, as well as immune population. And these are ongoing work. There is a lot of differences between the different disease manifestation, the more diffuse scleroderma, which has usually much more potent and acceleration of the pathology, versus the limited. And there is different ideas of how we think about generating, hopefully, what would be a more relevant treatment, which will have more effect. So this is one area which I wanted to share. And again, we went down to understand again what are the potentially relevant pathological cells, where they are located in the fibrotic tissue, what type of signaling is involved, and so on and so forth. So this is one part that I wanted to share. I think one lesson that is important, again, using the skin cell atlas together with this data that we acquired allowed us to really understand quite rapidly what are the dramatic changes, what is happening. And I think that kind of pushed us to think about not only the common disease, but going into rare disease and understanding them. And sometimes the biology of the rare disease can help us think as well about the common disease. So that's one area that's quite important, and we kind of identify these new cells that we call SCAF, or scleroderma associated fibroblast, and they seem to be quite important in coordinating the healthy and non-fibrotic skin. Another story or another lesson, that's the reason I'm bringing him here, was a study spearheaded by Björn Kragstein, who was really passionate to understand one very important physiological cue, and that is how erythrocytes, we produce 2 to 3 million erythrocytes per second, how are they regulated? And we know erythropoietin plays a very important role in actually regulating the amount of erythrocyte we are producing. And there's been evolution around these cells and pathways in Tibetans, which go into high altitude. But when we went deeply into the literature, we understood that unlike many of them, or maybe all of the important physiological cues, such as generating insulin by beta cells and so on, till today, we don't understand actually what are the cells that make erythropoietin and what is the underlying biology around this. And obviously that erythropoietin or control of the blood cells is clinically very, very important and affects millions of people's lives. And this is a very simple scheme of what's going on, and I won't show you all the data today, but basically erythropoietin from most of the literature that we've read, and also we verified this both in mouse and human, is actually coming from somewhere in the kidney. And over the years, many different cells were suggested to be the cells that produce erythropoietin without any type of evidence. And so that was sort of what we set out to do. And this is just a figure showing you again, erythropoietin is produced at least the physiological level. If you remove the kidney, the erythropoietin is not produced to sufficient enough. And so in adults, mainly produced in the kidney, and also it has this very abrupt induction upon hypoxia. So it's induced and then decreases very rapidly and goes to a new steady state. All right, so how do you actually find such, I would say, rare and I would say cells that are not been found using the standard way that we make atlases? So the first thing we did is go and start collaborating with the people in the field and identify tools that may help. And one of them was Roland Wenger, who is working on this area for 30 years. And he designed a very important tool, a mouse with a reporter that reports on these production of EPO, and by that label these cells. And then we use this tool to identify the cells that Bjork named Norn cells. Norn are the goddesses, female goddesses that decide the past, present, and future of a human being according to their religion. And we believe these cells are have this importance of maintaining our viability. At any case, they have a very unique feature. They express unique pathways for regulation and control of oxygen levels and obviously produce EPO. Again, there's the paper was published, and I encourage you to read it. And again, some of the sort of interesting and exciting results there is actually Norn cells are there at all times, okay? They produce EPO only upon a reduction, or produce the high levels of EPO only when there is hypoxia or one goes to high altitude or exercise and so on. But they're there and maintain their identity. Their identity is fixed, and the only thing that is changing upon hypoxia is a very small gene model. And that basically then allows us to actually search for them in mouse across development, across evolution, in humans, and so forth, so on and so forth. And indeed, we found the human equivalent of the Norn cells. We validated actually in quite a challenging way, because even if you look at kidney atlases, both mouse and human, you would find the Norn cells, but they would not produce EPO. And only if you really challenge the cells with hypoxia, you would see the EPO production. And we could basically find this only when we took samples from people who died out of CO poisoning, and unfortunately happens quite frequently during winter in Europe. Anyway, I'll finish here. Again, there are a lot of shared pathways and regulation between the Norn cells and the human and mouse Norn cells, and this is ongoing research to study them, to understand how to generate them in vitro in organoid, and then potentially use them or the relevant pathway therapeutically. Again, a lot of work we did on how they're regulated, and that's quite important, how they sense oxygen and then activate the relevant response to oxygen deprivation. So I'll stop here. Again, a lot of lessons, but I think the important one, thinking about the atlases, is there are likely going to be many examples where we're not going to fully understand the functionality of the cells, and sometimes looking the other way around, looking from the function side and then searching in animal models or in disease perturbation may help us pinpoint the cells and then go back to the atlases and then identify them at large. So that, I think, is another important lesson I wanted to share. And how much time do I have?
Ido, you still have 7 minutes.
Okay, good.
7.
All right, so I'll try to share in the last 7 minutes some new technology. We are always excited about new technologies. New technologies have been a driving force for the field. I'm overlaying some of them that I think that people in this community are very familiar with. But we thought one type of measurement is really missing. And I think, one aspect that we have a challenge with is can be seen from this sort of imagery. So if I would ask you what Rafael Nadal is feeling right now, I think people would have a challenge to say, but most would probably say he's suffering from a big loss. But in reality, if you look at this across time, what you see is that actually this is him really enjoying a great victory in the US Open. Okay, so if you think about it, everything that lives actually has a register for time. And this is the one point that our fixed atlases are perhaps missing, and identifying the different trajectory the cells are undergoing may really open a window for us to understand much of the biology of the atlases we make. And of course, there have been approaches, technologies, new algorithms that attempt to do this. For example, velocity, which looks at the splicing ratio to try to understand the trajectory of cells. However, these data are quite difficult without having a ground truth to kind of train our models. And as everyone knows who uses such data, changing the gene set that one works with can really change the outcome of these trajectories. So how do you solve this? Again, I'll make a long story short. So the work or the technology that we call Zmansek, and Zman in Hebrew and also in Persian mean time, allows us to actually have a empirical measurement of time by introducing labeled antibodies into the circulation. And then when we, at the end of the experiment, take out the tissue and process it, we would have on every cell not only its transcriptional identity, but also a timestamp of when the cell entered the tissue. And that allows us to learn a whole lot of information that we couldn't learn before. And we're, by the way, making a version of this technology, which it will be compatible. The whole thing was done on FACS-based technology that we have custom, but we are going to have hopefully shortly a method for commercial platform like 10x and so on. Anyway, there's a lot of different aspects of the technology that we had to develop and improve. I won't have time now to go over it. I promise Christine I'll stay on time. But anyway, it allows us to, again, build really a very significant understanding of cellular trajectory, or actually the empirical changes that cells are undergoing when they move from one area to another, from circulation to any one of the tissue. This is, for example, data from gut and lung. I'll just go quickly on some tumor data and more specifically on glioblastoma, which is a very difficult to treat tumor. And again, one of the challenges with this tumor, it really has a very dysfunctional immune microenvironment, which prevents immunotherapy treatments. And we used sort of the Zman-seq methodology to try to understand what are the signals that are changing the immune compartment there. And this is basically how sort of Zman-seq experiments are done. We here in this case, we added the GL261 glioblastoma tumor model. And then once the tumor grew to sufficient size, then we started injecting our Zman labels or time labels, and then sacrificed the mouse, and then took out the tumor, processed, and then we start learning about having not only the transcription information, these are myeloid cells and these are NK and T cells, but we also have a understanding of time. And here I'm really just projecting the data on this map. This is not the formal analysis. We built a formal analysis for this technology. But what you can see is basically the shift that the cells are undergoing with time, from these naive monocytes, which are entering from circulation, to these inhibitory tumor-associated macrophages. Again, a lot of data we learn on the different immune subsets, how they are changing. I won't go too much into this. But what I want to say, and I think that's your take-home message, time is such an important aspect of immunology, but I think in general in physiology, and we actually were underappreciative of how important this bit of information of this, and how it helps us understand the biology of immune cells in different tumors and different tissues. Just to mention, the way we analyze it is using the MetaCell algorithm developed initially by the group of Amos Tanai. So we break down the data into coherent groups of cells called metacells. And now for these metacells, we have the enrichment of various time labels we have in the data. And that allows us for every such metacell to compute when these cells enter the specific tissue or tumor, and how they were affected. And obviously we have all the molecular data associated to this. For example, this is the NK data, and we can basically follow when these cells enter the tissue and their exact trajectory, how they're changing over time. A lot of lesson of how they initiate a dysfunctional program when they enter the tumor from the periphery, and how they lose their cytotoxic activity and so on, and what are the pathways associated with this. So this is a very important learning information. And the last bit of information I'll show you, I think I'm putting too much for this time, but I just wanted to highlight this technology, is what we learn about myeloid cell changes in the tumor. And again, one specific focus was on TREM2. As I showed you, it highlights an important macrophage population, which is changing from the monocytes that enter into the periphery to the inhibitory macrophages that are basically differentiating in the tumor. And therefore, we used an antibody that blocks TREM2 activity, and now we combine the TREM2 blocking with the control antibody. And what we basically see, and this is your myeloid map, these are the most inhibitory macrophages or suppressive macrophages that you have in the tumor, and these are more inflammatory macrophages. These are monocytes that enter in, and different dendritic cell population. Bottom line is that what is happening if we don't, or we treat a control antibody, the cell will become these inhibitory macrophages, and with the TREM2 treatment, they shift to these much more inflammatory macrophages. This is the normal trajectory if we don't block TREM2, so the cells are undergoing this specific trajectory. It has to do with different signaling that is going on in the tumor, which is very interesting. And what we see is when we treat with TREM2, it completely blocks the transition of this differentiation of monocytes in the tumor, and they transition to much more inflammatory macrophages. Some of the molecules that are changing, so these are different molecules associated to inflammation and co-stimulation if we block the TREM2 pathway. And much of this suppressive module is not turned on if we block TREM2, which include PD-L1, IL-18 binding protein, and so on and so forth. And the beautiful thing around it is we can now to start to understand actually what are the signals that are activated in the macrophages in the normal sort of normal, if you will, changes when the monocyte enter the tumor. For example, midway, there's a very strong type 1 interference signaling that is completely blocked if we activate TREM2. So again, I don't have much time to go with, but just I thought it's important to highlight this. So ZMANSEQ is a new technology to record single-cell RNA-seq in time. It allows us to define the dynamics of immune cell in GBM and understanding better our anti-TREM2 antagonistic molecules. So I think I'll finish here to not go over time. Really thank the amazing people who did the work. So this is the scleroderma team, the Norn team, and this is the many people who contributed to the ZMANSEQ technology, which was spearheaded by Daniel Kirschenbaum, that I didn't credit. So thank you for listening.
Thank you very much, Ido. Christine, if you can start my video, please. Thank you. Again, thank you so much, Ido. Just as we wait for any questions that the audience might have, I have a couple of questions. One is just from this very interesting discovery of these Norn cells. So what level of understanding at the single cell level of the pathways at the transcriptional level are usually required to start designing like assays for perturbation? Because these cells are not as well characterized as all of the other classic cells. So I'm just curious how much level of understanding of the pathways and the co-expression networks are required.
Yeah, that's a great question. I think some of the I think the beauty of the HCA and where it's taking us is into actually developing new models. So you find a new subset of cells, and now you know, and many of them are let's say generated through many different physiological signals. So without actually making or replicating these exact cues, you would never have the appropriate cell type. So in order to produce Norn cells, actually, we're starting to understand the different signals and different transcription factors that are important to generate these cells, and which are a subset of fibroblasts, although they're actually very distant from fibroblasts. So now we're generating all types of models in order to generate them, or generate, let's say, similar cells with the capacity to generate EPO and many of the, let's say, signaling and sensors that these cells have for oxygen, and then that allows you to study their function. So I think, you know, the HCA is taking us to new journeys of new biology, and that's part, I think, critical part of the fun of discovery. That then now you start to develop new models. These new models take you to new types of direction that you didn't anticipate before. It's the same in the scleroderma for this new coordinating scout cells that we identified there, and their importance in maintaining the skin healthy.
So my second question is about just the state of integration of TCR and BCR with transcriptional and epigenetic states for immune cells, because that's an important aspect that complements the functional, epigenetic and transcriptional state of immune cells.
Yeah, absolutely. I mean, I think there's a very increasing importance of understanding how the TCR and BCR or their chronality is relevant for disease. It's obvious for all the hematological malignancies, which are, B cell derived or T cell derived, that, but, it goes beyond the autoimmune disease, obviously, this is very relevant. And I think there are quite good technologies these days to both look in the bulk level, which is helpful, as well as on the single cell level of such TCR and BCR, and overlay them on the transcriptional maps.
All right. I don't think we have any questions from the audience. So again, thank you so much, Ido, for this nice overview of the
Absolutely.
field.
We have a shy audience today.
Yes, it's and we have people from like different time zones as well. So great shout out to our colleagues in Boston, because it's almost in there. So
absolutely.
Yes, yeah. Great. So I think we will stop here, and so we're gonna take a 15-minute break. Thank you so much for joining this first very interesting talks. And so to join the next session, you go back to the agenda page, and then you can just click on join now at the top of the screen. So we're gonna resume again in 15 minutes. Thank you so much.
Bye-bye.